Professor Yang, and I'm a scientist. I'm here today to tell you a little bit about my profession. There are many different kinds of scientists, like physicians, mathematicians, astronomers, and I'm a genetic scientist. Genetics is the field that studies our DNA, our molecule of heredity. And how I did get here? I studied biology in a university. After that, I got a master's degree and a PhD in genetics. It's a long way. Today, I'm teaching genetics at the university, so I'm a teacher and also a researcher. Today, I'm going to show you some of our research and also research of other labs. My group and I currently evaluating the effects of the pollutants in water using fish as a experimental model. We simulate contamination with certain drugs and agrotoxicants in an aquarium with several fish. And after that, we analyze the effects of these pollutants in a period of exposure. We examined the blood and the organs with these animals with the microscope and biochemical tests. Come on, I will show you. A DNA molecule in our body is a teeny in our cells. And we use the fishless model like this. They are fixed fish that we already used in our research. Now I show you some slides with the blood of these fishes. Come on. This is a microscope that we use to show you in this screen the cells with the blood of the fishes. Here we can see a lot of cells with nucleus, and here we analyze the alterations. Here we have another image from our researches and how we can detect the impact of the pollutants in these organisms.
So let's understand how this experiment works. First, we choose the target pollutant to be studied, for example, a pollutant called X. Then we can put it in contact with the fish in different percentages, such as 25 or 50%, to evaluate the activity of the compound. In this experiment, at the same time, there will be fish living in clean water and others in contaminated water, as in tanks B and C. After a period of exposure, for example 1 month, the fish are then sacrificed to analyze their blood and organs. At this part, it's important to notice that the fish don't suffer. The entire protocol of the Animal Research Ethics Committee is followed, and the animals are anesthetized before euthanasia. Furthermore, the cells are prepared and transferred to glass slides so that they can be analyzed under microscope. And in the end, we have our cells slides that can be seen under the microscope. We can see their nucleus in a pink and purple color. We can also see their chromosomes, and all of it will depend on how we manage our cells until the final results. And looking to the cells through the microscope, we can see certain type of alterations, and we can compare the cellular changes that occurred in the fish due to the action of the pollutant in different concentrations. And By evaluating the final data of our analysis, we can establish safe exposure doses for the environment. And this is how Professor Eliane's research takes place in the animal cytogenetics and mutagenesis laboratory, but other types are also performed in our department. So, hello everyone, my name is Myllena and I'm currently a master's student associated with the Environmental Biotechnology program at the same university, and I'm here to show you other types of research areas that you can pursue that may be associated with genetics. So, firstly, we have the Biological Control and Insect Bioprospecting Laboratory. Here, researchers can study the pests that run agricultural crops and those that can be even associated with forensics. And continuing in this direction of the insect studies, they can also research the ones that can benefit ecology, medicine, and pharmacy. So, another one is the Microbial Biotechnology Laboratory, where they study different types of microorganisms such as endophytes, those that can be found inside plants and don't cause damage to it. Other studies are related to the selection of agents for bioremediation, which means that these organisms can help in the degradation of molecules that are difficult to clean from the environment. And they can also work in obtaining nanobiocompounds and also in the selection of agents that are resistant to radiation and genetically modified organisms. In addition, we have a laboratory related to animal genetics and biotechnology, in which we have insects such as bees being used as instruments for toxicology to understand their resistance to insecticides and even their genetic diversity. They can also be used to study gene expression and the selection of queen bees for honey production. The Plant Genetics and Biotechnology Laboratory works with the cell culture of plant tissues, such as those from the cereals pyrovianus, and with their production of metabolites. In addition, there are also studies on the productions of soma clones from the species and the genetic diversity promoted by the in vitro culture of different tissues. And last but not least, we have the Environmental Monitoring and Mutagenesis Laboratory, in which I participated, where we research basically the toxic genetic effects of different molecules and drugs on cells and other organisms, such as mice or even the cell roots of Allium cepa. I would like to pay a little more attention to the animal cell culture, where we take cells considered normal or tumorals from different animals, such as mice or even humans, and test them in a controlled space to assess the toxic effect of a certain compound. After the culture, we can see different types of DNA damage through some tests such as the COMET assay or the MTT, and through this type of study, which is considered across the world to approve initial research on living beings, we can begin to understand whether a compound may be beneficial or not for later use. So, these are some of the areas of research you can work with in the biotechnology, and I'm sure there's a lot more to be explored in other fields. So keep searching, and I'm sure you'll find something that might spark your interest in being a scientist in the future. Just keep in mind that research work is very detailed and the science needs to be dedicated and studied constantly. If you like our work, please find out more about the other research being done at our university.
And finally, we have a project to talk to the population about the university research and answer questions about genetics. The project is called Dr Genetica, that means Dr Genetics. And this beautiful chromosome is our mascot. I will show you some of our work here.
It's market day, Wednesday, Friday, no matter the market. It's market day, anyone who wants to can come. When we think of genetics, the first term that comes to mind is DNA. But have you ever heard of allele, gene, chromosome, and are a little confused when it comes to differentiating? Let's understand. Think of our DNA as a cookbook and divide it into chapters. Each chromosome is one of these chapters. They organize themselves in the cell nucleus and represent the most condensed structure of the genetic material, which can be observed in the metastasis phase of the cycle. Because with condensed material, correct division in cells is more likely to occur properly. Humans have 46 chromosomes arranged in pairs. 22 pairs are autosomes, and a pair represents the sex chromosomes, known in the XY system for some organisms. And what is a gene? Think that within these chapters there are several specific recipes that give the instructions for our body. Each of these recipes is a gene. Genes determine characteristics such as eye color, hair color, and even resistance to some diseases. That is, a section of DNA that controls a certain trait. For example, the TP53 gene, known as the guardian of the genome. This gene produces a protein that helps suppress tumors, preventing the growth of cells with damaged DNA. Mutations in this gene can favor the formation of tumors. And the alleles? They are different versions of the same gene. They are the ingredients that change the final result. Each person inherits two alleles for each gene, one from his father and one from his mother. If these alleles are the same, we call them homozygous. This means that they will give the same information, such as blue eyes. But if they are different, such as one for blue eyes and one for brown eyes, they are called heterozygous. If only one gene was responsible for this, you could receive a blue-eyed allele from your mother and a brown-eyed allele from your father. In this case, the dominant allele is the one that would define the final color of your eyes. But what do you mean eye color is not determined by a single gene? Good. Some of our features,